Hi everybody, this is Jill Morgan and I want to talk to you today about the lower airways. This is in Jardins, Chapter 1, Part 2. So the lower airways are below the larynx. They are part of the tracheobronchial tree whose main function is to conduct air. So this shows the conducting zone and the respiratory zone. The conducting zone goes from the trachea all the way through to the terminal bronchioles. The respiratory zone goes from the respiratory bronchioles all the way through to the alveolus.
The structures of the lungs. So there are, again, here are the conducting zone and the respiratory zone, but now you can see the generations, 1 through 28. Now, the numbers between the subsegmental bronchi and the velar sacs is not really known. This is a best guess on the part of physiologists. However, I want you to note that airways with cartilage are all bigger than 1 millimeter. Airways that do not have cartilage are less than a millimeter. The generations 1 through 9 all have cartilage that goes all the way through the subsegmental bronchi, from the trachea to the subsegmental bronchi. Bronchioles and terminal bronchioles that are greater than 1 have cartilage. Bronchioles and terminal bronchioles that are less than 1 millimeter do not. And they branch from the terminal bronchioles into the very, very tiny respiratory bronchioles where we start to see some gas exchange.
So what zone are the cartilaginous airways in? Anybody want to take a guess? They are in the conducting zone. If you said conducting zone, good for you, kudos. Another question. Which airways compose the non-cartilaginous airways? Can you remember? The bronchioles and terminal bronchioles, they're less than 1 millimeter in diameter. The lower airways consist of the bronchi, large airways, the small airways, the bronchioles, the epithelium that covers them, which is a ciliated, pseudostratified columnar epithelium. Each little cilia is about 5 to 7 millimeters in length, and there are about 200 on each basal cell. It also is part of the lamina propria, which kind of holds everything together and contains a bunch of stuff. It's got connective tissue, mucous membrane sitting on top of it, nerves, glands, vasculature, like arteries, and smooth muscle, and mast cells. And then there's the cartilage. Cartilage in the bigger airways, up right until the bronchi and bronchioles that are less than a millimeter. Those have no cartilage, but the ones that are greater than a millimeter also have cartilage in them to keep them rigid, so air can move in and out.
So this is the histology of the tracheobronchial tree. It has a great picture of an alveolus and shows you where it is in the lung, and then the lamina propria in the bottom corner. I encourage you to look at this picture in your book and the parts. We're going to talk specifically now about the epithelial lining of the tracheobronchial tree. As you can see, it's made up of 4 different layers. The top layer is the mucous blanket, which we'll talk more about, the epithelium and the lamina propria. And then on the bottom, you've got the cartilage layer. Pictured in the middle in the lamina propria, you have a submucosal gland, which makes up about 95% of the sol layer. It's mostly water. And then smooth muscle right below a basement membrane. The little basal cells that you see on top of the basement membrane and the cilia on top of those are your ciliated pseudostratified columnar epithelium that line the bigger airways.
So I want to talk a little bit about cystic fibrosis and about the mucus that can get stuck in the airway from cystic fibrosis. The pathology includes three things: a bunch of secretions in the airway, and those lead to accumulated airway secretions that get thick and sticky. And if there's a partial obstruction, then there can be air trapping. And if there's a full obstruction, it can create atelectasis or even hyperventilation. So those little alveoli can become over distended and don't work. They don't snap back into place. They break apart. They don't work very well. There also can be some airway remodeling, meaning that the airways that with all this thick, sticky mucus in it that's hardened can become disshaped and don't work anymore.
In this picture, you see mast cells in the lamina propria. A cross-section, this is a cross-section of the bronchus showing those mast cells. And as you know, when the mast cells disintegrate, they release a bunch of different chemicals. Well, why is that important to you? Because in asthma, an antigen comes along, stimulates one of those mast cells, they break apart, they bust open with all those chemicals, and that makes those smooth muscles contract.
This is a video that I'd really like you to watch. It talks about immunologic mechanisms of the airway. It's a great representation and well worth the time. It's in your Blackboard.
There are two types of immune responses. There's cellular immunity and there's humoral immunity. The cellular immunity involves sensitized lymphocytes that are responsible for tissue rejection in transplants. This immune response also is termed type 4 or delayed or a type of hypersensitivity. The humoral immune response involves circulating antibodies that are involved in allergic responses such as allergic asthma. That's what we're talking about with this picture. Antibodies, also called immunoglobulins, are serum globulins or proteins that defend against invading environmental antigens, such as you see pictured here. Antigens can be pollen, animal dander, feathers, dust mites, and they come in from the environment and are identified as IgG, IgA, IgM, IgD, and IgE. The IgE reanogenic antibody is basic to the allergic response. The mechanism of the Ig antibody antigen reaction is when a susceptible individual is exposed to an antigen, like dust mites, the lymphoid tissues release specific IgE antibodies. Those IgE antibodies travel through the bloodstream and attach to the surface receptors on the mast cells. It's estimated that there's between 100,000 and 500,000 IgE receptor sites on the surface of each mast cell. Gosh, that's a lot. Once the IgE antibodies attach to the mast cell, the individual, or more specifically the mast cell, is said to be sensitive to that antigen. So really it's not the first time the antigen attaches, it's the second time a person gets exposed to it that this reaction happens. Once you have a sensitized mast cell, the antigen comes around again and it makes the mast cell disintegrate. So it releases all types of chemical mediators. Some of those mediators are histamine, heparin, platelet activating factor or PAF, eosinophilic chemotactic factor of anaphylaxis or EC-A, and leukotrienes. The release of these chemical mediators causes increased vascular permeability, allowing liquid into the airway, smooth muscle contraction, so it squeezes the airway, and then increased mucus secretion and vasodilation with edema. So the inside of the airway swells up and you get a lot more mucus in there.
The trachea extends from the carina all the way to the level of the second costal cartilage or fifth thoracic vertebrae, and then divides into the right and left main stem bronchi. I want you to note that the bifurcation of these two is the carina. So, larynx to carina is the length of the trachea. The carina is located about the fifth thoracic vertebrae, and there are about 15 to 20 rings of cartilage. All of them are C-shaped in adults. In a child, carina are omega-shaped, and they're the smallest part of the airway. The first generation or main stem bronchi are the right and left that you see highlighted in red, and they lead into the right and left lungs. The cartilage is greater than 1 millimeter. So all of these airways right now that we're talking about have cartilage in them. So going down a little further, we can see the bifurcation into the lobes of the lung here, here, and here. 3 on the right and 2 on the left. The subsegmental bronchi end the airways with cartilage in it. So, what is, in centimeters, what is the average diameter of the adult trachea? It's the size of a quarter, or 1.5 to 2.5 centimeters. What's the term for that point where the right and left main stem bronchi bifurcate? It's really important because we want to be above that point when we intubate someone. So super important to respiratory therapists. So what's that called? Can you remember? Well, it's called the carina. So I want to talk a little bit about the mucus blanket. It has 2 layers, the gel layer that sits on top and is thick and sticky, and the sol layer, which is very watery. In fact, it's 90 to 95% water. The cilia live in the sol layer, and the entire blanket of the sol layer is made up by submucosal glands. The gel layer is formulated by the goblet cells. Question: what is the term for the very viscous or thick and sticky layer of the mucus blanket? Is it the gel layer or the sol layer? Did everybody shout it out? It is A, the gel layer, the layer that traps bacteria and sits on top. So let's talk for a moment about what happens to this mucus blanket when you smoke. So in the picture, you can see that a non-smoker has an open airway. They've got strong, healthy cilia that wave back and forth, and they have clear fluid for the sol layer. Now, and the muscles are relaxed and open, and the airway is open. However, in the smoker, you have a very irritated, inflamed bronchial wall. You have thick, sticky mucus layer, and then you have a very narrow airway. It's narrowed with inflammation and with secretions and with tightening of those little muscles around the bronchial tubes. Air can get trapped and can actually damage the alveoli or air sacs. Thank you for your time and attention. It's been great to be with you. Have a great day. Bye.